On this week's Arabidopsis Research Roundup, I'm delighted to be joined by Dr. Miriam Gifford from the University of Warwick, and we're going to be talking about a paper which is going to be published in Plant Cell, and this is a large-scale biology paper, the title of which is Changes in Gene Expression in Space and Time Orchestrate Environmentally Mediated Shaping of Root Architecture. So as it's a large-scale biology paper, there's a massive load of data here, so I don't think we're going to pick through this in extreme detail, but hopefully Miriam is going to be able to give us a great overview to this paper. So thank you, Miriam, for joining us, and as I say, it would be great if you could give us an overview to the paper. Thanks.
Great. Well, thank you. Nice to be able to explain a bit more of our, as you said, enormous dataset paper. I think the key things here is that we try to investigate how changes in the environment that affect root development are regulated by gene expression changes in different cell types over time. Mm-hmm. Here, what we did was try and address some questions that are often definitions in textbooks. So what a cell type is, cell type identity, and thinking then to take that original textbook definition and instead look to see how the environment shapes cell identity, and then how these cell identity responses are actually come together to enable a whole root system to adapt to or change in response to the environment. Okay.
So can you talk us through the main technique that you used in the paper?
The main technique that we used in the paper was fluorescence activated cell sorting, or FACS, which I have been using for some time now because it's a fantastic way to isolate individual cell types of a multicellular system in order that we can analyze molecular changes within those cell types. So we grew Arabidopsis seedlings on vertical plates to look at root architecture, and then isolated those cell types using FACS and used gene expression analysis in response to changing the environment that the roots were grown on. So we were able to look at treatments and cell types and time all in one data set.
Okay, so this is kind of a technique which was pioneered by the Benfey lab in the mid-2000s, and so in this data set, can you explain the cell files that you looked at specifically? Obviously, the root has a lot of different cell files, but you just focused on a couple of those, I think.
Absolutely, as you said, it was a seminal method developed some years ago, and there's a lot of work come from being able to isolate different cell types. So other groups look at different species or also different types of development. Here we focused on the pericycle, for that's a cell type in which there's dedifferentiation and development cell division, enabling formation of new lateral roots. And so that was the main cell type of interest. And then as a comparison, the cell type that's overlying the pericycle, closer to the environment, which is the cortex. There's another reason for that, which is this is part of another large piece of work from my group where we're looking at nodulation in legumes. And during nodulation in legumes, it's the cortex from which there's new cell division. So I'm interested long-term in comparing different cell types that have a similar developmental ability.
Okay, so you've given a little bit of a clue there, but about the sort of treatments you use on these roots. But so you made two main treatments in this paper. So can you explain the treatments that you use, and maybe an overview of what you saw between the two different cell files, if that's possible in a short time?
Yeah, I'll give you the highlights. Yeah. So we used responses or treatment of nitrogen. Okay. To which has long been of interest in my group, looking at the changes in root architecture. So input of nitrogen activates or stimulates lateral root development and emergence. And the second treatment is actually a biotic treatment, rhizobia. And now again, I've explained this link from nodulation to Arabidopsis, so that's a part of a long-term question understanding in my group. Here we use rhizobia, and we find, even with Arabidopsis that's a non-host, obviously there's no nodulation, but there are changes in root architecture which we think are not just a defense response against this biotic input, and actually are regulating root development.
Okay. And so you mentioned about using the two cell files, one of which is closest to the environment and one which is closer to the vascular tissue of the plant. So I think you see interesting changes when you look at the interaction with the rhizobia and how those two different cell files respond, kind of in a temporal way. So can you talk a little bit about that?
The temporal aspects were really interesting. Now, I knew that we would see differences between cell types, some of which are probably entrained by cell type identity. Others, I think, are to do with location. And, of course, in plants we know that location or position affects identity. More specifically, though, as well as differences between the cell types, we actually did see temporal dynamics, and we found, in general, responses occurring earlier in the cortex, a more outer cell type, than in the pericycle. Okay. So you did being able to look over a high resolution time series, we could see evidence for an outside to inside response. We don't yet know if this is a direct outside-inside response or what the precise molecular pathways are, but we have a lot of nice new clues to follow up on in the new data. Okay,
so you mentioned that there's a really detailed time scale there, and just to highlight that, they take data on 0 time points and then 1, 2, 4, 6, 8, 10, 12, 14, 16, 20, 24, 36, and 48 hours. So when you're adding to the fact that you've got these two treatments as well, all of those time points, then it starts to bring together the enormous amount of data, enormous amount of gene expression which has been analysed in this paper that you've all managed to put together, and somehow have come out with some coherent ideas as well, which is excellent, I must say. So I've got a question about the up and down nature of the root. Let's say, maybe I'm not sure if this is unfair or not. So we know that as the cell files move back up the root, then they will change their identity a little bit. With this paper, though, you're taking an average, I guess, of every position at the root. So are you looking in future work to try and discriminate a little between cells closer to the tip and those further back at all? Is that something for the future, maybe?
Absolutely, that's a perfect description of what we would do next. Okay. You're right, with the FACS or with the cell sorting, so far we've isolated cells in a cell file, which therefore do have a range of developmental ages. We know that for pericycle we are enriching for cells that are progenitors of the lateral roots, but we still have a range of developmental ages, and one of the parts of work we're starting in my group now is looking much more at cell by cell, and we're using a variety of single cell RNA sequencing methods that enable us to isolate and then sequence cells and then reconstruct, using the sequence information, their position within the root. Okay. So this should allow us to start to look even finer detail. I'm sure we're going to find even more genes that are differentially expressed to do with space, time in terms of hours, but also developmental time or age. Okay, cool. And so I think that will, of course, then have far-reaching implications for understanding a lot more about how developmental and molecular processes are integrated.
Okay, so I've got a couple more questions just to finish, and this first one might be a little unfair perhaps, considering this is a really great paper, but is it possible using this technology that you could look at the cell files in the same plant? So you could have a GFP tag and like an RFP tag, so a different colour that could be sorted separately, and that would be the true comparison then, whereas instead of you're comparing transgenic plants which grown separately and then comparing that data, but comparing the data from the same plant? Is that a possibility for the future?
Absolutely, and we already have plants where we've combined different fluorophores. Okay. And I think for looking cell by cell, that would be much more useful. Okay. When we're comparing cell file to cell file, perhaps that specificity of the same plant origin is less of a biological variation, but certainly if we want to compare cell to cell, more like a really true in situ for gene expression, then using the same individual plant becomes much more important. And also, though, now the methods for cell-by-cell sequencing are much better, then I think that becomes feasible and therefore useful. We don't just want to generate a whole load of more data. We actually want to learn something about plant development and cell identity.
Absolutely. So finally, as I said, there's a lot of data and there's a lot of information about gene expression temporally and spatially. So a lot of people will be interested in this. So where can people find this data? So I assume it's all freely available, and where can people take a look for their gene of interests?
There are lots of places. So we have, as you said, it is a huge data set, and we have the data from the paper available on the Plant Cell website, which links actually to Dryad, which contains the large-scale data sets, including the gene expression networks and the files, so people can look up not just gene by gene, but actually what the links between genes are. And we also have gene expression data available on the EFP browser for gene expression, which enables you then, of course, to look at genes across different data sets too.
Yeah, absolutely. Great. Okay, so thank you very much, Miriam, for having a chat about this. And as I said, you alluded to the fact there's lots of other interesting stuff going on in the lab, so we'll look forward to hearing from you about those soon enough as well. Thanks so much.
Great, thank you.